pleased to introduce as well is Dr. Stephanie Sherman. She obtained her PhD in human genetics and is currently a professor in the Department of Human Genetics at Emory University in Atlanta. She has focused her research on understanding genetic and environmental factors that are associated with the clinical conditions associated with Down syndrome. She and her colleagues across the U.S. have established a large clinical research program to identify biological pathways that are perturbed in Down syndrome, and the goal is to identify possible interventions to reduce the effects of the extra chromosome 21 and increase the quality of life for those with Down syndrome. So really pleased that Dr. Sherman's here with us again today. And I'm just excited that y'all are getting the opportunity just to hear about studies in our area, because I think a lot of times, you know, these are so close by that they're definitely doable for a lot of our population. So,
Great.
Sherman.
Thank you.
Put that on.
Yeah, let's see.
And
Okay.
if you have a pocket, you can put that on.
Yeah. Great.
And this will get you through the presentation.
There we go. Great, thank
Thank
you. First, I want to thank Cam and Catherine for setting this up, and also Vince, who was very involved in helping us get some money to do this, and very involved in just the process of bringing research awareness to this group. So I am going to talk about just our way. Well, let me step back. What you just heard about the clinical trials and interventions are based on the science. What interventions do you do? And so our work has really started to try to look at the basic science to ask, where do we go? What kinds of interventions can we use? And what is the empirical evidence to say that would be a good intervention? So that's what I'm going to talk about today. How do you identify the altered biological systems that are particular to individuals with Down syndrome, and then identify possible treatment interventions for those? Okay, so first, I mean, everybody knows this. Everyone with Down syndrome has an extra chromosome 21. That's a given. That's part of the diagnosis. But every individual is unique. And it's that aspect of uniqueness that we work with in order to try to identify these pathways. So what do I mean by that? In what ways are individuals unique? And we can even start at the fetal life, because really the majority of fetuses with Down syndrome don't make it to term. The majority are lost because of aspects of survival. So understanding who survives to term and who doesn't is an important thing. Why are 80% of the fetuses lost during pregnancy? When they do come to term, why do some have significant birth defects, but others have no birth defects at all? They have structurally normal heart, intestinal system. And then with respect to cognitive and behavior, I mean, I bet everybody in this room understands the spectrum that you see on both aspects, how well the individual learns with trisomy 21, and then also the behavioral aspects. For example, why do some individuals with Down syndrome have autism spectrum disorder and others don't at all? And then lastly, the aging process among individuals with Down syndrome. Again, a very wide spectrum. Although many have the pathology related to Alzheimer's, for example, the onset of the behavioral aspects of Alzheimer's varies considerably. Some might have an earlier onset, but others might have a very late onset. And so understanding why that's true, understanding the subgroups, as you said, why do some respond to the drugs and others don't? So that's what we're trying to find, the underpinnings for those subgroups. Okay, so again, what contributes to the uniqueness of an individual? Or better said, let me say, what contributes to the severity of the clinical outcomes? Because that's what we're most interested in. And here I've just listed, all the different things that you're familiar with that are associated with Down syndrome at a higher frequency than what you see in the general population. Again, congenital heart defects, these other defects of the gastrointestinal system. And then, again, the variation that we see in cognitive performance, behavior, and aspects of dementia. So big profiles, a lot to learn about why you have an effect of an extra chromosome 21. So what are the reasons for? What contributes to this uniqueness? It's the roll of the dice. Sometimes it's just chance. Things coming together, and they lead to either protection or susceptibility. Clearly, the DNA involved and the variants associated with everybody's genome bring a big feature to an individual's spectrum of disorders. And then lastly, the environment. So chance, genetic variance, and the environment all play a role in that uniqueness of an individual. So that's what we try to tease apart. So here's just the general way that we see this problem. And here, I'm going to talk about Down Syndrome 360 as kind of the overall umbrella project that we like to consider, because we like to really consider all these aspects at one time. So this is an effective, a cost-effective program, and I'll try to explain why, that identifies factors that are really involved to reduce the clinical effects of that extra chromosome 21. So we examine environmental factors, clearly, genetic factors, and through this process of characterizing that information in each person, then we can identify these altered biological pathways. That's the whole idea. And once we can understand this, then we can start to develop interventions in a very empirical way. So a very targeted way, for those with an extra chromosome 21. And that's exactly what Roche built on, that the one drug that you talked about. They understand that there's a difference in the neurotransmission. There's an excitatory component, and there's an inhibitory component of anybody's neurotransmission system. In Down syndrome, there's just too much of the inhibitory. So this drug starts to rebalance things and bring it back into balance, and then hopefully with an excellent outcome. Okay. And we think that it's also important not only just to function on one aspect, one component of Down syndrome. So for example, not only cognition or not only behavior, but really all aspects. For the following reason, I mean, here's just a list of, here's some potential medical conditions that might exist among an individual with Down syndrome. And then quality of life. We know, for example, that for congenital heart defects, if an individual has a congenital heart defect, has it repaired, that later in life they have some cognitive problems, difference in cognitive trajectory, and then later in life cognitive problems. And that's true with or without Down syndrome. Okay, that's a known phenomenon. But we also know if you have a congenital heart defect, then you increase the number of hospitalizations, which also makes you susceptible to other conditions, again, reducing quality of life. We know it reduces activity level, and then we know that it increases issues related to controlling weight. All these are important quality of life issues that we want to see, improved for the whole individual. So again, taking a holistic view of the person and capturing those data in order to ask what aspects are really, or what biological aspects are altered so that we can intervene. Okay, so here are two projects that we have been involved in. And I want to say, and that we're trying to roll together and put all the data into the bigger Down Syndrome 360 umbrella. And I want to really point out that this takes a huge collaboration among not just clinicians, among researchers, and among the families. And you know how, as we've been saying this morning, it's a lot of effort on your part. And we really appreciate that, because it couldn't be done. I mean, we would get nowhere in terms of trying to achieve these goals. So we, here I'm talking about doing the holistic thing. Well, we've never gotten money to do the holistic thing. So we have done components. So I'm going to talk about the Heart Project in a minute. This is one that we have is most mature in a sense. It's the Cognition project that we're recruiting for, so I'll talk a bit about that too. But the important thing is, for both these projects, and you can't see in the little writing, but it's a multi-site project, we have the same protocol, essentially, so we can pull the data together and continue to have this, the sample size grow. So in the data collection, we involve the mother in terms of getting her maternal health and pregnancy history. We ask for the medical records of the individual with Down syndrome to pool all the information about what went on at birth, what went on later in life. And then for the cognition test, we do a battery of the neuropsychological tests, many of the ones that were spoken of with Sarah and Jane Ann. We also ask for blood from the individuals with Down syndrome, if they can provide it. Sometimes now we can do with saliva. And then we ask again for blood or saliva from the parent. So we get that trio, the mother, father, and the child, if we can, in order to do our genetic studies to correlate with the environmental studies. As I said, it's a multi-site project, and these are just some of the locations of groups that have been involved in doing these kinds of assessments. So it's an exciting group, and we hope to make that grow as we grow resources. Okay, so let me talk a little bit about the Down Syndrome Heart Project, just to give you an idea of of the pattern that we use, or in a sense, the research protocol, and then what kind of outcome that we expect. So again, among those without Down syndrome, about 1% at birth have a heart defect. Among the kids with trisomy 21, about 50% have heart defects. Now, I just want to make sure that this is really clear, because 50% have structurally normal hearts. And what is that? And what's going on there? Among those 50% that do have heart defects, the majority are these septal defects, but each one has a different kind of underlying cause. So atrioventricular, AVSD is sometimes called hole in the heart. So all four chambers are communicating with each other because the structure have broken down within there, have not developed properly. Atrial septal defect, again, the upper two chambers, and ventricular septal defect. Now, we focused on atrial septal defect for the following reason. Among those without Down syndrome, it occurs in about 1 in 10,000. Again, in individuals with the trisomy 21, it occurs in 20%. So that's a 2,000-fold increased risk for this very specific kind of heart defect and very severe heart defect. This has to be repaired, around the time of birth, as soon as the baby is healthy enough to tolerate an operation. So it's a very important thing to understand. And again, there's multiple operations that have to occur. So it's a life-changing event. Okay, so how do we do this? How do we go about this? We try to do an exhaustive search of the genetic risks for this AVSD. So we have two groups, essentially. Our cases and controls are defined as those with Down syndrome and a complete AVSD that again takes it to a little bit more homogeneous subset, which is helpful in these kinds of studies. So those are our cases. And the controls are individuals with Down syndrome and structurally normal hearts, as documented by echocardiograms. So now, at this point, we have about 300 of each one of those groups. And those are the comparisons that we make, and I'll show it a bit, some of the results. But then we have, because of our collaborations with Roger Reeves at Johns Hopkins, we're able to take what we find in these clinical research studies among individuals, among humans, and then take those genetic variants that we think are high priority, that really stand out to be more frequent in this group than in the other, and then put it into a mouse model system. And why is that important? Because that then starts to tell us the function of that genetic variant. What is happening, and is it really important? We have many genetic variants. All of us have a ton. I mean a ton. So trying to find that one that's a high priority and moving it into a model system like mice before we start to think about it in humans is a very important, cost-effective way to really prioritize these variants. And so Roger works with two types of mice, at least, two categories. Here I'm just showing the indication of they have representations of 2 chromosome 21, so the typical chromosome constitution, and then the other mouse that has 3 chromosome 21s. And what he can do is take the variants that we identify in this part of the study and stick it in this mouse and say, okay, what heart defects are present, or any heart defects? And then do the same thing. Take that same variant and put it in a background of a mouse that has 3 chromosome 21s. And again, ask, is it exacerbated? Do we see more pups with heart defects, or is it the same and it really makes no difference? So a big step before we move further into other kinds of trials. Okay, so as I said, we have about 300 of cases and 300 of controls. What have we learned so far? In terms of environmental factors, we know 3 things. We know that this particular kind of defect is more prevalent in females than males among children with Down syndrome. We know there's some risk factor, though when we talk about race, ethnicity, it's very hard to understand what that's measuring. But we know that there's twice the frequency of kids with Down syndrome born to those self-identifying as black compared to whites, and half as many for those self-identifying as Hispanic or Latino. So something is going on there very importantly, and it's trying, the next step for us is to uncover that. And then lastly, we know that folic acid supplementation around the time of heart development, so that's about 8 weeks of pregnancy, plays a role. So if there's a lack of that, there's an increased risk of that heart defect. In terms of genetic factors, so on the other side, what do we know? We thought this was going to be a very simple problem. When we went into this thinking, and we went into NIH saying, this is going to be easy. A 2,000-fold increased risk, it's got to be something big, a common genetic variant that only is unmasked on the background of a trisomy 21. We were so wrong. It's like every other complex disorder. What we're finding is there's a lot of rare variants that are playing a role and increase the risk for AVSD. Interestingly, those rare variants are some of the same ones that they're finding among kids that have these heart defects but don't have trisomy 21. So that's important. What we learn in Down syndrome can extend to the general population. So where are we with it, trying to work out these pathways? And we know we have to look more at folate metabolism in our next studies. We now know that the cilium is the coolest organelle. So on every cell, there's like this antenna, that's up there saying, oh, I shouldn't do this on a recording. I'll keep going. Oh, there's an antenna up here saying, where am I? What am I supposed to be doing? What's the environment? And it's called the cilium. And that signaling system, that organ that says, okay, now I know where I am, now I have to do something, seems to be perturbed, or the genes that have the variants in it and look potentially damaging involve these primary cilia. So again, and the important thing is that they've seen this in the mouse model too. So now we have a pathway that we can pursue. And then lastly, the VEGF pathway. This is a growth factor pathway, and I won't go into the details except to give you now an example of how we've taken one of the genes that we found that is in that pathway and moved it into the model system. So I just want to show you this as an example. So deleting a gene from this VEGF pathway, it's called CREL-D1, that's not important at this point, increases the risk of CHC when it's on a trisomy background. So let me just show you the outline here. So here's the mice, has 2 chromosome 21 representations. A plus means that it has 2 copies of the CREL gene, so that's what you expect normally. And neither this one or when you delete 1 copy of the CREL1 gene presents with heart defects. So none of the mice have an increased susceptibility of that. Let's take the trisomic mouse, okay, the Down syndrome model. Again, not perturbing this other gene, but in that one, just by having an extra chromosome 21, 5% of the pups have heart defects. But if I don't know if you can see this, but if you delete 1 copy of that CREL-D1 gene, then 1/3 of the pups have heart defects. So indeed, when you put that variant on a trisomic background, you do see an increased susceptibility. So this is one that now is a very high priority to move on and ask, what is the function of this gene and how can we pursue that altered pathway? 5 minutes, okay, I gotta go. Okay, so understanding factors that lead to the variation in cognition. This is the project that we're recruiting for. I'll be at the back, and if you're interested in it, actually, I gave Vince some contact information, so you can let me know if we can call you and tell you about it. But let me just give, again, an idea. We're looking at the variation of cognition and behavior. This is funded by the Lou Mind they just changed their name, sorry, the Lumine Research Down Syndrome Foundation. Very good support. We wouldn't be able to do this without their money, so I want to thank them much. Same type of thing. So this is the protocol. Right now we have 250 participants, and as Sarah and Jane Ann said, this is not enough to really understand what genetic factors are associated with this, but it's a good start. And this one is not a case control, but we can look at a cognitive outcome. We can look at one test related to, say, working memory or learning, and we can look at that whole distribution and then ask the question, what's associated with explaining that variation that we see among individuals. Right now, we don't have funding to do the genetic component. We are just building that foundation so we can take the samples and we can store them. But what we can do is we're doing all the other aspects of looking at the medical records and everything to define birth defects, days in NICU, hospitalization, and then associate these with high performers, low performers, and really ask, does this impact the cognitive and behavioral outcomes? So that's where we are at this point. So where do we want to be? We want to be in this mode of the Down Syndrome 360. We want to get to a point where we have 1,000 participants, 1,000 families investing in this, in time and effort, and do 1,000 whole genomes. So a whole genome, why do that? It's the basic biology of each of us. And it's the simplest thing to capture and characterize, and it's very easy to share in the whole community. And once it's done, it's done. You know, it's not going to change. So it's such a resource, such a gold mine in terms of identifying risk factors. Where are we with the recruitment network? We're really trying to expand, not just with us, but now we're talking with Lund Downs. This is a great project that's run out of the UK, and they're focused on Alzheimer's, but they're collecting many of the same data points that we are in the Down syndrome cognition study. Down Syndrome Connect, which you'll hear next, helps facilitate the recruitment, but also we're working with them to say, can we use some of the data, again, with permission from the families and the individuals with Down syndrome, to use in this database to connect with the genetic material. It's going to be we have to be very specific about what data do we collect among all the sites that we're hoping will join this study. So we're working with a new entity called the Trisomy 21 Research Society. This is an international group now of investigators, and they have a clinical committee that's beginning to identify which are the core clinical aspects that we want to capture among these 1,000 individuals. In terms of biological samples, we could collect everything from you. We could get urine, we could get skin, we're sticking right now with saying, let's just get blood or saliva if the child can't tolerate the blood sample. And as I said, do the whole genome sequencing. Where are we with that? Well, I have to say, we're in the infancy in terms of getting the investigators together. So we have just started talking about an international Down syndrome genomic consortium. People have now agreed to start to put their data together. This is such an exciting thing. I would say that the Down syndrome community is a bit slow on this, coming together as a consortium. This was a huge step that just happened about 3 weeks ago when there was a Keystone meeting and the investigators said, are we going to do precision medicine for individuals with Down syndrome? And if the answer is yes, we have to start pooling our resources. So this, I think, is very exciting. I'm very Pollyannish, too, so the others are not as excited as I am, but I know we can do this. I do know we can do this, and it is going to be a push from families. It has to be grassroots to say this needs to be done. Okay, where am I? I have to get off my soapbox. Okay, but not only are we going to help the individuals with Down syndrome, but I also believe that we're going to help the wider community. As I showed you in the heart results, we can extend, those are extendable. What we find in Down syndrome was overlapping with what we find in heart defects. And I believe this is true for everything that we find. And you'll hear now a big emphasis on this aspect, this whole idea of Alzheimer's, and understanding that individuals with an extra chromosome 21 are at a high risk. We can use what we learn from them to understand the whole lifespan of what contributes to potential dementia later in life. So it's an exciting time. As this whole idea of where are we in research for Down syndrome, right on the horizon of finding interventions. And so I'm very excited. I'm very excited to talk to you all about it. Oh, and here's just the ton of people that work. I have to pull out Tracy Rosser, who has worked putting this together. She couldn't make it this time, but she's our director of all the recruitment and of course. And Lumine. Thank you. Thank you.